Targeted cancer therapies thus far have used genomic alterations and differences in gene expression to predict how successful therapy will be. However, the things that determine a specific response to changes in metabolism are complicated and involve both genetic and environmental factors. The Warburg Effect, or aerobic glycolysis, the process discussed in class that converts glucose to pyruvate in the cytoplasm of cells, is used as a diagnostic for tumor cells that predicts cancer outcome. In this process, most cancer cells predominantly produce energy by a high rate of glycolysis followed by lactic acid formation in the cytosol, whereas in most normal cells, most energy is formed with a relatively low rate of glycolysis followed by the pyruvate oxidation in the mitochondrial matrix. Liberty et al. Assert that partial inhibition of a glycolytic enzyme with selectivity against tumors is needed in cancer therapies because glucose metabolism is required in nearly all bodily tissues, so full target inhibition and disruption of glycolysis throughout the body would be disastrous for the human body. This can be done with competitive inhibition as seen with this glycolytic enzyme because an inhibitor binds directly to the active site of the enzyme and prevents substrates from binding to the enzyme, which greatly reduces the enzyme's function. Metabolic control analysis was used to identify how differences in flux control would allow for targeting the Warburg effect, which would make great strides in identifying therapeutic responses for cancer therapy. They did this through analyzing flux control coefficients and reaction-free energies on lactase production for each step of glycolysis. It is known that steps with high FCCs and low delta Gs exert more control over flux in the pathway, so the FCC and delta G were compared to each enzymatic step in glycolysis. In this analysis, it was found that only GAPDH's flux control increases with the presence of the Warburg effect, so it must exert the strongest control during the Warburg effect. When inhibited, there is a reduction in glycolytic flux, so this information can be used to target the Warburg effect. Next, different GAPDH inhibitors were evaluated for their toxicity. Conegonic acid, Ka, was found to be the only inhibitor that can cause decreased flux through competitive inhibition of the GAPDH enzyme. Tests confirmed that the target of Ka is GAPDH and by association the Warburg effect because only glucose uptake, Spearman correlation R equals 0.6 and P equals 9.4 times 10 to the negative 7, and lactate exertion, Spearman correlation R equals 0.6, P equals 5.6 times 10 to the negative 7, were predicted of Ka response under a survey of roughly 50,000 molecular and physical variables as shown in this diagram. The sensitivity of highly glycolytic cancer cells to Ka was tested by using estrogen-responsive breast cancer in mice. GAPDH activity was reduced and glycolysis interrupted. Furthermore, researchers discovered a window within 24 hours of injection where Ka was most effective in targeting the Warburg effect, decreasing 3pG and PEP concentrations within 5 to 10 minutes of treatment. The data also indicated that Ka is bioavailable, tolerable at certain doses, and can induce serious changes in the glycolytic network of tumors. Pyruvate and lactate were found to be elevated in tumors but remained mostly unchanged in the liver, plasma, and muscle. These results show that Ka spares normal tissues and exerts bioactivity mainly in tumors. So partial GAPDH inhibition by the competitive inhibition via Ka can be tolerated in most healthy cells while exhibiting selective toxicity in tumors. As shown here, the healthy cell has little flux control compared to the cell undergoing Warburg effect, which has large flux control. Despite both being targeted by Ka, only the cell undergoing Warburg effect was affected by Ka and flux control was increased where needed, limiting negative effects on healthy cells. The important conclusions from this research are that Ka exerts many metabolic disturbances across various pathways in the mitochondria that are each targets in cancer, which makes it a good candidate for cancer therapy and a new way to view cancer research as a whole.